Good afternoon, students. Today we are going to study the next topic, ultrastructure of a eukaryotic nucleus. Right? So clearly I have written eukaryotic nucleus. Why not prokaryotic? Because prokaryotic cells do not contain a well-developed nucleus, not the nuclear membrane. Okay. So eukaryotic nucleus. For the very first time, the eukaryotic nucleus for the very first time was discovered by the scientist Robert Brown in 1831. Right? So in 1831, Robert Brown was one of the first scientists who for the very first time generally discovered the eukaryotic nucleus. Right? Now, nucleus is generally called as the controlling center of the cell or director of the cell. Why it is called controlling center or the director of the cell? Because the entire functioning of the cell is generally regulated only by the nucleus. That's why it is generally called controlling center of the cell or the director of the cell. Now, nucleus, right? When you talk about the ultrastructure of the nucleus, we will discuss about the shape. First, the shape of the nucleus depends upon the shape of the cell, right? The nucleus might be spherical, it might be oval shape, it might be disc shape, sometimes it is bilobed, sometimes it is multilobed, etc. Size. The size of the nucleus also depends upon the size of the cell. Now, if a single nucleus, we all know that a single nucleus is generally present in a cell. So, if a single nucleus is present, we generally called it as uninucleated cell. If two nucleus are present, we generally called it as binucleated cell. If more than two are present, then we generally called it as multinucleated or polynucleated cell. Now, polynucleated cell present in the plant is generally called as coinocyte cell, right? And polynucleated cell present in the animal is generally called as syncytial cell. So syncytial is a polynucleated cell present in animal, and coinocyte is a polynucleated cell present in the plant cell. So here we come across the ultrastructure of a eukaryotic nucleus. It is made up of four major components. You can see the first is called as nuclear membrane, second is called nucleoplasm, third one is called nucleolus, and the fourth one is called chromatic reticulum. The entire nucleus is generally bounded by a double layer membrane. That double layer membrane is generally called nuclear membrane or nuclear envelope, right? Okay, so nuclear membrane is also called as karyotheca. Why thicker? Because it is a thick membrane. Now, as it is a double layer membrane, one is the outer membrane and another one is an inner membrane. The space present between the outer and inner membrane, can you see this? Yes. The space present between the outer and inner membrane is generally called as perinuclear space. Okay, so perinuclear space is a space present between the outer and the inner membrane. Now, in between the nuclear membrane, there are small minute aperture or the pore. This pore are called nuclear pore, and they are generally surrounded by a dense ring-like structure. This dense ring-like structure is generally called as annulus or annuli. Right? This is nuclear membrane. In between them, the small pores are present called nuclear pores, and they are generally surrounded by a dense ring-like structure called as annuli. This is outer membrane. This is inner nuclear membrane. Outer nuclear membrane will be attached to the endoplasmic reticulum. Okay. Outer membrane will be attached to the endoplasmic reticulum, whereas the inner nuclear membrane will be attached to the dense network called as fibrous lamina. Right? Fibrous lamina for the very first time was been discovered by the scientists Harris and James. Harris and James discovered the fibrous lamina. That's what all about your nuclear membrane. What would be the function? Nuclear membrane generally separates the nuclear material from the cytoplasm of the cell, right? Cell के अंदर cytoplasm रहता है, right? Okay, and this is your nucleus. So nuclear membrane separates the nuclear material from the surrounding cytoplasm of the cell. Chalo, second one, nucleoplasm. The entire nucleus contains transparent, homogeneous, semi-solid acidophilic matrix, and that matrix is generally called as nucleoplasm, right? You can see the transparent, homogeneous, jelly-like acidophilic matrix is present, granular matrix is present, which is called nucleoplasm. Nucleoplasm is also called as karyolymph, right, or the nuclear sap. Now this nucleoplasm contains two important components. One is the nucleolus and the second is the chromatin reticulum. So nucleoplasm generally contains your nucleolus and the chromatin reticulum, right? And also nucleolus contains nucleic acid, that is DNA and RNA, proteins, enzyme and mineral, right? So protein hai, enzyme hai, mineral hai, your nucleic acids are there, they are present in the entire nucleoplasm, correct? The third component, nucleolus. Nucleolus is a small spherical acidophilic body present at the periphery of the nucleus. Is this present at the center of the nucleus? No, it is present at the side, corner, that is periphery of the nucleus. So it is generally a spherical body, acidophilic body present at the periphery of the nucleus. Nucleolus for the very first time was discovered by the scientist Fontana. Fontana was the first scientist who for the very first time discovered the nucleolus. Now, it might be a large size nucleus or a small size nucleus. Nucleolus, sorry. It might be a large size nucleoli or a small size nucleoli. Large size nucleoli is generally present in the oocyte, that is the ovum or the egg cell. Correct? Okay. So neurons and the secretory cell. Whereas the small size nucleoli is present in the sperm cell, the muscle cell, the blastomeric cell, etc. Right? So that is all about your nucleolus. And also nucleolus contains nucleolar associated chromatin, fibrils, मैट्रिक्स एंड ग्रैन्यूल. ये न्यूक्लियोलस के अंदर उसी का न्यूक्लियोलार एसोसिएटेड क्रोमैटिन मतलब रेटिकुलम प्रेजेंट रहेगा, उसका मैट्रिक्स प्रेजेंट रहेगा, ग्रैन्यूल्स प्रेजेंट रहेगा एंड फाइब्रिल्स present rahega. That's what all about your nucleolus, a small spherical body discovered by the scientist Fontana. And the last one, the fourth one is chromatin reticulum, right? क्या आप देख सकते हो पूरे आपके न्यूक्लियस में एक coiled structure scattered है. So the entire nucleoplasm, right, contains your thin coiled thread-like structure. This thin thread-like coiled-like structure is generally called as chromatin reticulum or chromatin network. Now this chromatin network or reticulum contains chromatin material, that is either the DNA or the RNA as a genetic material. Now DNA is made up of two types of protein, histone protein and non-histone protein, right? Histone protein is required for the protein synthesis. Now on the basis of this chromatin reticulum, there are two different types of chromatin threads which are being seen in the nucleus, right? First is called as heterochromatin and second is called as euchromatin, right? Euchromatin is a lightly stained chromatin, right? And heterochromatin is a darkly stained chromatin. Euchromatin contains large amount of your DNA and a less amount of RNA, but heterochromatin contains larger amount of RNA and lesser amount of DNA. That is all about your heterochromatin, which is the darkly stained chromatin, and euchromatin, which is the lightly stained chromatin. See, chromosome or the chromatin has an ability to absorb the stain. Those who are absorbed lightly are called euchromatin. Those who are absorbed darkly are called as heterochromatin. And also, this your chromatin reticulum generally contains a repeated units, and this repeated units are generally called as nucleosome. So nucleosome are the repeated units which are generally found in the chromatin reticulum. This is the ultrastructure of the eukaryotic nucleus. How many components? 4 components. First is nuclear membrane, also called nuclear envelope or karyotheca. Second is nucleoplasm, also called as karyolymph or nuclear sac. Third is nucleolus, and fourth is chromatin reticulum. Thank you.